But chapter 5 is about the working cell. So last week we learned about all the cell parts, the mitochondria, the nucleus, the Golgi apparatus, all that good stuff, all of the cell parts. And this week we're going to learn how those parts work in certain ways. So cells control their chemical environment, both inside and out. They have to use energy to do it. They also use enzymes. Enzymes are used for everything. And they use their plasma membrane to move stuff in and out of the cell. So part 1 is about energy. Cellular respiration, chemical energy harvested from fuel molecules, get broken down and then built back up again. And throughout this process is how our cells generate energy for work. And work is mostly growth, our cells growing or dividing, replicating. Our cells need energy to go through mitosis. Energy makes the world go round, but exactly what is energy? It's defined as the capacity to cause change. Some forms of energy are used to perform work. Energy is the ability to rearrange a collection of matter. When we talked about chemical reactions, we talked about how the breaking and reforming of chemical bonds is what actually generates energy. All of our cells in our body do this for us. It is not something that we consciously do. Our body just does it, like our heart. Our heart doesn't consciously beat. We don't tell our heart beat, beat, beat every second of every day. It just does it automatically. Our cells generate energy automatically because they need to. Potential energy, there's two types of energy. The first is potential energy. It's stored in an object. It's stored energy that an object has because of its location and or structure. Think of food. Once we eat, we convert that food into energy. But until we actually eat it and break it down, that energy is just stored in the chemical bonds of the carbohydrates, proteins, and lipids. So an apple sitting on a shelf has that particular chemical structure that you see at the bottom, but it's not going to actually generate energy until we eat it and our body starts breaking those bonds. So that is called potential energy. Kinetic energy is the energy of motion. Our cells convert the chemical energy stored in food molecules into kinetic energy that takes the form of muscle movement, among other things that our bodies do for us. Kinetic energy is actually being used and not just stored in some molecules somewhere. Most of the time, our kinetic energy goes to moving our muscles however we're doing it. Even sitting up straight, you're moving muscles. Even if you're not moving, you have muscles in your spine that are still holding you up and using that energy. Your heart is still beating. That's a muscle that uses energy constantly. Conservation of energy. So both machines and organisms can transform kinetic energy to potential energy and vice versa. In all such energy transformations, the total energy is conserved. Energy cannot be created or destroyed. This is a law of thermodynamics, a scientific law. Energy just gets converted from one form to another. The principle is called conservation of energy. If you ever take physics, you'll get into this a lot more. For now, we just know that energy takes different forms, cannot be created or destroyed. Heat. If energy cannot be destroyed, where does it go? Every energy conversion releases some randomized energy in the form of heat. Heat is a type of kinetic energy contained in a random motion of atoms and molecules. It's a product of all energy conversions, but it is difficult to harness. You think of catching energy, it's not that easy. Catching heat that comes off the fire. You can't really catch that, it just kind of goes. Heat is energy in its most chaotic form. It's the energy of aimless molecular movement. And this is what allows us to maintain our body temperature. Energy gets converted in our muscles use it, but as a byproduct, we generate heat, and it keeps our body temperature steady. Entropy. Scientists use the term entropy as a measure of disorder or randomness in a system. All energy conversions increase the entropy of the universe. Molecules store varying amounts of potential energy in the arrangement of their atoms, specifically in their bonds. It's like the apple we were talking about. And you can see its molecular formula at the bottom. This chemical energy can be released when the bonds are broken during a chemical reaction. Organic compounds are relatively rich in this chemical energy. And remember, organic just means made of carbon. Carbohydrates, lipids, proteins, those are organic molecules in and of themselves. They don't have to be made naturally or without pesticides. If they have carbon, they are already organic. And once we eat them, our body breaks it down, breaks those bonds, rearranges them to incorporate them into our body systems or our body structures. And that releases energy. Cellular respiration is one such chemical reaction, and there's two parts to this reaction. Part one is the chemical breakdown of the food molecules that we eat, carbs, lipids, proteins. And then part two is the storage of that energy in a form the cell can use to perform work. And we store that energy as a molecule called ATP. Humans convert about 34% of the energy in food to useful work, such as the contraction of muscles, which leaves 66% of the energy released by the breakdown of fuel molecules, generates our body heat, maintains our internal temperature, 98.6 all the time. Whether we're standing in 115-degree weather or we're standing in 20-degree weather, our internal body temperature stays the same. Calories. We're going to talk about the science definition, not the food definition when you look on the label. The actual science definition. 1 calorie is the amount of energy that can raise the temperature of 1 gram of water by 1 degree Celsius. So you can actually measure this. You burn food under a container of water to turn the stored chemical energy into heat, and then you measure the temperature increase of the water. Food calories are actually kilocalories. And the energy of calories in food is burned off by many activities. Even sleeping burns calories. Just a little bit, not a lot, but pretty much every action you take burns some calories. Of course, the more calories you burn, that's when you lose weight. ATP is short for adenosine triphosphate. It's the actual molecule that our bodies use for energy. The breaking of bonds during chemical reactions generates these molecules for our bodies. And it acts like an energy shuttle that stores energy obtained from food and then releases it as needed at a later time. The structure of ATP consists of an organic molecule called adenine, that's supposed to say adenine actually, plus a tail of 3 phosphate groups. It gets broken down into ADP and a phosphate group when it's time to release energy for the body. And that extra phosphate group is what energizes our molecules and cells to perform mechanical, chemical, or transport work. There's a video there. We're not going to watch it right now, but if you want to learn a little more about ATP, that would be the video to watch. Plasma membrane interactions is the next category of cellular work that we're going to talk about. We talked about the structure of the plasma membrane last time. All the little circles, the heads and tails of the phospholipids. The big purple things are for transport, proteins. And you have the carbohydrates, the ID tags, and then the cholesterol woven throughout it to make it stronger. But we're going to talk about the big purple things that were in that picture. Those are proteins, and they're used for transport. So cells must control the flow of materials to and from their internal environment. And they do this via the many proteins that are embedded within their membranes. Transport proteins are located within membranes and help move substances across a cell membrane. It kind of acts like an open doorway, or sometimes it's a closed doorway that needs to be opened, and that's where the energy comes in. Passive transport. Cell membranes are selectively permeable, only allowing certain substances to pass. Passive transport is a diffusion of a substance across a membrane without the input of energy. So you can see in the picture, there's a membrane in the middle. And the tiny little molecules, which are likely water, just kind of float through. And it mainly has to do with size. They're small enough to get through. So they don't need an open doorway. They just can go straight through the phospholipids. But the green molecules, whatever they are, they're too big. If we need green molecules to get in or out of our cell, that's when the transport proteins come in. Passive transport molecules contain heat energy that causes them to vibrate and wander randomly, like we talked about the fire, the energy that comes off that fire and how it's hard to catch. Diffusion is the movement of those molecules so that they spread out evenly into the available space. It does not use energy. It's still a form of passive transport. And you think of it like smells. Somebody's in a corner of a room and they spray perfume, the people immediately around them are going to smell that perfume. But eventually, those perfume molecules will diffuse into the air, and that circle that can smell that perfume gets wider and wider and wider. During passive transport, we say that substances diffuse down their concentration gradient. That's a phrase we use. The concentration gradient is the gradual difference in concentration of a dissolved substance in a solution between a region of high density and one of lower density. So food dye is a good one. If you dip or if you drop a few drops of food dye into a glass of water, it starts out really, really concentrated, but as you stir it, those dye molecules spread throughout. So there's an even amount of dye molecules in whatever that substance is. Those dye molecules started out really concentrated and then they spread out to where they were not. So that's called diffusing down their concentration gradient. Some substances do not cross membranes spontaneously, or they may cross very slowly. Like I said, it has to do with size. Water molecules are small enough that they can go in and out. But some substances are a little bigger. They need a little help. So these can be transported via facilitated diffusion. To facilitate means to help. So these are just molecules that need a little extra help getting into or out of the cell. Those specific transport proteins act as open doorways. There's still no energy input needed. These doorways are just open. Molecules that are small enough just float across the membrane via the open doorway. Doesn't take any energy to keep them open. They're just open. Osmosis is the diffusion of water across a selectively permeable membrane. And you can see the blue water molecules are water. The red line in the middle is some kind of membrane. And the water molecules want to spread out into the area that doesn't have any water. The water is moving down its concentration gradient, because it's spreading out into an area where there is no water. So if you compare a solution, we're going to use Kool-Aid as an example. These three vocabulary words: hypertonic, hypotonic, and isotonic. Hyper, if you think of a person that's hyper, they have too much energy. A hyper person always has too much energy. Children, small children are usually like that. You always say, oh, that kid's so hyper. So hyper just means high, like a high concentration. So a hypertonic solution has a higher concentration of solute. You think of Kool-Aid, we have sugar, we have Kool-Aid, we have water. Those are the three substances used to make Kool-Aid. So in a hypertonic Kool-Aid solution, we would have too much sugar and not enough water. A hypotonic solution, hypo would be the opposite, has a lower concentration of solute, so not enough sugar and too much water. And then an isotonic solution has an equal concentration. It's just right. The taste is just right. So we can apply these concepts to actual scientific solutions. Osmosis plays a huge role in water balance in living things. Osmoregulation is the control of water balance within a cell or an organism. In our bodies, we have our urinary system, our kidneys, our bladder. If you drink a lot of water, what do? You have to pee a lot. That's your body working to get rid of excess water. Plants have rigid cell walls made of a lot of wax that will keep water in. Plants like water, they want extra water, just in case there's a drought. Plant cells are healthiest in a hypotonic environment. They want more water than solutes. It keeps their walled cells turgid. This is another word for stiff. When plants start wilting, so you know they don't have enough water, that is the opposite. So here's a couple examples. In an isotonic solution, animal cells, which is us, and that red disc is a blood cell. Our cells like isotonic solutions. We like it when water and solutes are even on both sides. Plant cells, not so much. They need the extra water. They need a hypertonic solution. I'm sorry, a hypotonic solution where there's more water. If there's not enough water, like I said, it wilts. It dies. However, in a hypotonic solution, when there's too much water, our blood cells will burst or lyse. But plant cells like it. They like a lot of water. And then in a hypertonic solution, there's too many solutes and not enough water. Our blood cells shrink and shrivel and they don't work properly. And in a plant cell, the plasma membrane will actually peel away from the cell wall. That plant cell needs a lot of water to maintain their shape. If there's not enough water, it shrivels and shrinks and the cell membrane peels away. So as a plant cell loses water in that last picture, in a hypertonic solution, it shrivels and its plasma membrane pulls away from the cell wall in the process of plasmolysis, which will usually kill the cell. You can see in the GIF, the plant cells start off really full of purple. You can see the cell wall kind of shrinks, or the cell membrane shrinks and pulls that purple away from the cell wall. And the plant will eventually die. Which means they're not getting enough water. Active transport, the pumping of molecules across membranes. It requires that a cell expend energy to move molecules across that membrane. Exocytosis, exo, exit means out, is the secretion of large molecules within transport vesicles. So expelling, exit, exocytosis, pushing stuff out that it doesn't need. And then endocytosis, endo means in. This is taking material in via those vesicles that bud inward from the plasma membrane. And then there's a specific kind of endocytosis called phagocytosis, which translates to cellular eating, where a cell engulfs a particle and packages it within a food vacuole. So exocytosis is out, pushing stuff out that the cell does not need. Endocytosis is in. The plasma membrane buds around it and then brings it in. Cell signaling. The plasma membrane helps convey signals between cells and then between cells and their environment. Our cells are constantly talking to each other. They have to make our bodies function properly. Receptors on a cell surface trigger signal transduction pathways that will relay the signal and convert it to chemical forms that can function within the cell. It's kind of a domino effect. One cell sends a signal and then all the cells around it will send the signal to other cells or other body parts, or a bloodstream, or a brain. Signal transduction pathways are actually pretty complicated. We just have to know they send signals to cells to the surrounding tissues. All right, skip that one. Enzymes is the rest of chapter 5. Usually we do this in 2 parts, but since we're online, we're doing it all together. Metabolism is the total of all chemical reaction in an organism. Most metabolic reactions require the assistance of enzymes, proteins that will speed up those chemical reactions. And all living cells contain thousands of different enzymes, each promoting a different chemical reaction. Activation energy is a term that we use for a chemical reaction to begin or activate. Chemical bonds in the reactant molecules must be broken. This process requires the molecules absorb energy from their surroundings. So this energy is called activation energy. How much energy does it take to actually start or begin or activate a chemical reaction? Enzymes reduce the amount of activation energy required to break bonds of the reactant molecules, making reactions happen faster. So in the little graph at the bottom, the solid red line is the activation energy without an enzyme. So you have to build up this much energy before the reaction can start. Once you insert an enzyme into the equation, you have the dotted line where you only have to maintain this much energy for the reaction to start. So it lowers the enzymes lower the activation energy needed to start that chemical reaction, whatever the chemical reaction is. There's thousands of them that go on in our bodies. So we just went over that. The lock and key model. Enzymes are very selective in the reaction that it catalyzes. Like a lock recognizes only one shape of key, each enzyme recognizes only one substrate. The substrates are the specific reactant molecules. And the active site is the part of the enzyme where the enzyme and the substrate meet. So this is a useful picture because there's going to be some matching on your practice quizzes and on your lecture exam of the lock and key model. The key is the substrate, the lock is the enzyme, the place where they actually come together and meet is called the active site. And the end result of any chemical reaction are called products. You have the reactants, you have the products. Sometimes the active site will fit to the substrate and the enzyme will change shape a little bit. This interaction is called induced fit because the entry of the substrate induces the enzyme or makes the enzyme change shape a little bit. So this is just an overview of enzymes. You have the enzyme. In this case, it's called sucrase. Remember, enzymes end in ASE. And the active site is that top part there. It's ready for the substrate. The substrate is the key. In this case, it's sucrose. Sucrose is the carbohydrate or the sugar. And they come together and they bind. And then an actual chemical reaction happens. And you have the products that get released. And the products, when you split apart sucrose, are glucose and fructose. That's just an example of a chemical reaction between enzymes and substrates. Enzymes can function over and over again. They do not get used up during a chemical reaction. Our body just recycles them. Many enzymes are named for their substrates, but with an ASE ending, like we just saw. Sucrose got broken down by the enzyme sucrase, so it can get a little confusing, but if it's ASE, it's an enzyme. If it's OSE, it's a substrate or sugar. So remember, sugars end in ose, enzymes end in ace. Sucrose is the sugar, sucrase is the enzyme. Enzyme inhibitors. To inhibit means to block or stop. So to inhibit, restrain or prevent, there's a lot of synonyms for inhibit. Enzyme inhibitors can prevent metabolic reactions by either binding to the active site or near the active site, resulting in changes to the enzyme's shape so that the active site is no longer recognizable by the substrate. So it's like changing a lock on the door. If I'm going to come in with the same old key, it's not going to work. We change the shape of the lock. Or sometimes kids like to come through the hallways and stick gum in the keyhole. That would inhibit me from sticking my key in my keyhole and unlocking the door. That has happened before. So those are just examples. Enzyme and substrate binding normally. Enzyme inhibition by an imposter. So if the inhibitor comes in, blocks the active site, this would be like the gum in the keyhole. And then inhibition of an enzyme by a molecule that causes the active site to change shape. So this would be like changing the lock. My key doesn't work anymore because the lock has changed. Some products of a reaction may inhibit the enzyme required for its production. This is called feedback regulation, and it prevents the cell from wasting resources. So say we need something, so our body produces it for us. Once we have enough, that same product will stop this reaction and just turn it off and say, okay, we're done now. We don't need it anymore. There's a video about it if you'd like to learn more. Many beneficial drugs work by inhibiting enzymes. Penicillin blocks the active site of an enzyme that bacteria use in making cell walls. If bacteria doesn't have its cell wall, it's easier to kill. Ibuprofen inhibits an enzyme involved in sending pain signals. So when you take ibuprofen, Advil, Motrin, it's actually blocking whatever pain you're feeling. Many cancer drugs inhibit enzymes that promote cell division. Cancer is when cells grow out of control. There's ignoring the checkpoints, blazing straight through. So cancer drugs, we want to stop that process. So we do that by stopping the enzymes. Lab example, you guys are going to be doing a lab next week. An online lab, of course, but it's going to talk about catalase. Catalase is the specific enzyme that we're using during the lab. Like we said, most enzymes end in ACE, and this is the actual chemical reaction. So hydrogen peroxide in that big brown bottle, that stuff's called hydrogen peroxide. If you add catalase to it, it bubbles. And that bubbling is the catalase but H2O2 splitting. And you're making water, and those bubbles are oxygen. There will be more information on that coming.